Okay, Rayan again with Oceans for Everyone. In this video, I think we'll talk about tagging, tracking, and genetic sampling. All right, tagging involves attaching markers to marine animals for identification and monitoring to collect data on their movement, their behavior, population dynamics, things like that. All right, so you have external tags, internal tags, and then satellite tags as well. Like external tags would be like flipper tags, fin tags, and PIC tags, like passive integrated transponder tags. They're easily visible, low cost usually. They're used for fish, marine mammals, and turtles and things like that. Internal tags are usually acoustic tags or radio tags, and they're implanted or ingested, and they provide a bit more data and are less visible. All right, usually. Satellite tags like GPS or Argos tags are used for large animals and tracking long-range movements via satellite. They're good for migration studies, seasonal movements of species like whales, shark, turtles, things like that. Monitoring feeding habits, breeding areas, nurseries, social interactions. They're good for estimating population size or survival rates through recapture of tagged individuals, all kinds of different things. They're good for also identifying critical habitats, really, and informing management decisions, which is a big part of why we collect a lot of the data that we collect. But you kind of have to make sure that you're ensuring that the tags are securely attached without harming the animal, because tags may fall off or be damaged or lead into data loss. You know, you have to minimize the effect of tags on the animal's natural behavior and health. All right, and tracking involves the monitoring of the movement and the location of those tagged animals over time, and that way you can understand migration routes, habitat use, spatial ecology, things like that. We have acoustic tracking, satellite tracking, radar tracking that are, that come from the different types of tags that you use. So the acoustic tracking, like you would think, it emits sound pulses. It has underwater receivers that track the signal. We use this for like coastal and reef studies and fine scale movement patterns. Satellite tracking is a little different. It transmits data to satellites, tracks animals over large distances, so we can study long-range migration patterns and even ocean basin movements. Radar tracking, a little different again. It emits signals that you can track either by handheld or aerial receivers, and we use these for like short-range tracking, like estuarine or freshwater studies too. Just depends. With tracking, you can like study how species like sea turtles and sharks like navigate vast spaces of ocean. And you can identify breeding grounds, feeding areas, nurseries. You can investigate the impact of environmental changes, how climate change and human activities are affecting movement patterns. You can observe seasonal behaviors, daily behaviors sometimes, diving patterns in marine mammals, a lot. But you kind of have to make sure that you're getting reliable data transmission, especially like in deep areas or remote areas. You have to think about the battery life as well, the size of the tag and the capacity of the battery to make like really long-term tracking and to maximize the tracking duration. Genetic sampling, so connecting DNA samples from organisms to study their genetics. So you can understand diversity, population structure, and evolutionary history as well of these animals. So we'll take, more often than not, you'll take a tissue biopsy. You take a tissue sample to do a tissue biopsy. You can get environmental DNA as well. It's like directly from the water. And environmental DNA, where you collect water, or collect samples directly from the water is really cool, because you can analyze the DNA that's shed by organisms just as they exist in the water, and you can detect the presence of species. You can monitor biodiversity. It's really cool. You can analyze specific DNA sequences to identify variation. You can study gene flow and evolutionary relationships. You can confirming the presence of a species or even discovering a new species is amazing. Like, the party really pops off when a new species gets discovered. Is really cool. Conservation genetics as well. You can identify genetically distinct populations that need protection. Evolutionary studies, like I said, relationships and history among marine species. Sometimes there's difficulty in collecting samples from endangered species or elusive species. So there's that. Sometimes you spend a whole lot of time waiting to come across an animal just to have a shot at collecting its DNA. You also have to make sure you're avoiding contamination of samples with like non-target DNA. It's really complex too. Like, data interpretation is, especially for like mixed or like degraded samples, is like extremely complex. So these techniques are combined with each other a lot of the times for like really comprehensive research. Tagging and tracking provides like detailed movement and behavior data. With genetic sampling, you can get insight into population structure and connectivity. And when you're using all three, you can get a really holistic understanding of species life history, migration, conservation needs. It's really cool. Like, studying shark migration patterns using satellite tags and acoustic receivers, or studying like conservation efforts for sea turtles using genetic analysis to identify distinct populations of turtles. You can combine tagging, tracking, and genetic sampling to understand the connectivity of coral reef fish populations. They're really powerful tools that can give you insight into behavior, movement, and just diversity. They're crucial for informed conservation and management efforts across the world. All right, trying to keep it short. I think that's it. That's it on sampling, genetic sampling, and tagging and tracking. All right, thanks guys.